From Microbe TV, this is Immune, episode 102, recorded on February 11, 2026. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back. It is still really, really, really cold here.
But you know, it's getting here. Oh my gosh, it's 5 degrees C. It's tropical. It has been below zero for weeks. Also joining us from Cleveland, Ohio, Steph Langle.
Hi there. Yeah, my laugh maybe gave me away. I tend to not be able to hold in my laugh when you guys make me chuckle. So hello, good to see you. Hi, Cindy. It is tropical. It feels that way in Cleveland as well. It's above whenever it's above, the teens Fahrenheit. I'm like, whoa.
It has totally been below zero for the last month, 6 weeks, and today and yesterday it's now getting above zero during the day.
Yep.
Also joining us from Madison, New Jersey, Brienne Barker.
Hi, everyone. I am also very happy with this balmy weather. 39 Fahrenheit.
I mean, it's still cold, right?
Oh, it's still cold, but
our barometer is kind of screwed up now.
Yeah, this past weekend was so cold that
the
I'm pretty
humidity
happy.
Is very high, right? I have this thing showing humidity, and it was like 90% this morning. I don't know why it's so high.
Wet.
We should have some more of our Florida and California colleagues on. Our Texas colleagues.
Yeah, we're all in the cold.
We're all complaining.
I can go visit my parents. If we need it, I can go to Tucson.
Oh.
But I see people are now walking around in shorts. They think it's spring.
Well, I do have to say, when I walked outside yesterday afternoon, I was like, do I even need my coat? It's really warm out. And I look at the temperature, I'm like 39. That's not that warm. You do need you.
If you're outside for a while, you're going to feel cold.
Yeah.
Anyway, spring is 2 months away, right?
The groundhog, the stupid groundhog.
Groundhog.
It's all his fault.
We were talking about the groundhog on Daniel's program last week. And I said to Daniel, how do we know actually what the groundhog does? And he says there are people who guess, actually, because we never see the groundhog, right? Nobody sees any particular groundhog coming out and going back in. It's just...
And how do we know what the groundhog actually sees?
No, we don't.
We can't see inside the groundhog's
head. No. They pull Puxatawny Phil out, and then they're like, "Is it sunny?" Then he saw a shadow.
This is all just a cultural thing.
It is. Of course
it is a really silly thing.
I think you should look at the Farmer's Almanac, because that's a pretty good predictor of overall weather trends. The big picture.
Did they get it right this year?
This year, they said we're going to have a really bad winter here in the Northeast. And they were right. That was we had a lot of snow. It was cold. So a piece of paper, a book, maybe it's online now. I don't know. But
I would just like to say I'm going to knock on some kind of wood, because you said that in past tense, that it was bad. I hope that it is a past tense.
I think
we still have a ways to go here.
6 weeks. We have 6 weeks, right? We got the rest of February and then March. And then usually after March, it's it. You don't get snow anymore, right?
In Ithaca.
Well, it depends where you are, of course. Here in New York City area, although I do remember as a kid, one Easter it snowed, which is, I think that's in April, right? Easter is.
It is. Right? Once when I was a kid, it snowed on Mother's Day in upstate New York.
That's in May?
Mm-hmm.
Oh, my gosh.
I think the latest that it has ever snowed in Ithaca is in June. But I've not seen anything like that since I've been here. One benefit of global warming, I guess. Climate change. It's warmer.
I know. Anyway, I had somebody say to me one time, well, at least the kids, when they go trick-or-treating, it'll be warm. I'm like, okay, global weather catastrophe, but sure, our kids can go trick-or-treating when it's warm. I don't know if that's a great trade-off, but sure.
No.
So I was telling Brianna this morning, I was looking up the definition of banal, because I had heard a quote, which is the banality of evil, which is what the U.S. is going through now, right? And banal means lacking originality, freshness, or novelty, as in discussing the weather.
But listen, it's
I felt so bad.
To. Also, we don't get to see each other. We see each other only once a month. So there's going to be some.
I don't know what's wrong with discussing the weather. It kind of has a major impact on your life, right? Yeah. I mean, for farmers, they would discuss the weather, right?
Absolutely.
Very important.
So I don't know why it's banal.
I think we evolved to talk about the weather because we didn't have proper, places to be inside all day. So stuck with it.
Well, I don't know why we started doing it, but it was probably early on when I didn't know how to start, or something, and it just stuck. But I think it's interesting to hear how it's different in different places, and most listeners are okay with it. There are a few, I don't know, maybe 5% who don't like it.
They can skip through or put us at like 3x speed.
Yeah, you could do it.
Chipmunks talking about the weather.
But you know now that the algorithms don't make it sound like a chipmunk anymore.
Oh, really?
Yeah, they are digitally enhanced to go fast without that.
That's interesting.
It's very interesting. Yeah, so you can go on YouTube and play things faster, and the voices are normal.
That's true.
It's very cool. Technology
people.
Okay, today we have two very cool science stories. A snippet is from Steph first.
Great. So my snippet of science is in Science, the journal Science. The first author's name is Verena Lynch. And actually, let me double check to make sure there's not a co-first author situation so that
I don't think so. No, there's 1, 2, 3, 4. So I don't think so.
Okay, like it. Well, I don't. Either way, the only one, Lynch et al. And the senior author is Emma Slack. Actually, there's 2 stars. There's 2 corresponding authors. So to me, that would suggest a 2 senior author situation. Yep. Meredith Dyard and Emma Slack. And they are in Zurich, Switzerland, a couple other places, but I believe the main authors are in Zurich. And the title of this paper is Vaccine Enhanced Competition Permits Rational Bacterial Strain Replacement in the Gut. And I thought this paper was just super interesting and super cool. And why I came to it is I've been following Emma Slack's work for a while. Her lab, a little bit back, was one of the first to show that IgA can enchain individual bacterium to each other long ways. It can actually make them grow in like chains, or it can clump them together. And the outcomes of that are multifactorial, but you can imagine if you're clumping bacteria, you can get rid of them easier. You can not permit them to access the epithelium, keep them above the mucus and not through the mucus. But it also could be an evolutionary benefit to the bacterium, even to allow it to grow in a particular niche. So it's not always IgA blocking the bad guys, but it could be IgA shaping the microbial ecology. And other groups have shown that bugs are responding to IgA binding to them. And this idea of shaping the ecology of the microbiota. And so this paper brings this concept of vaccine enhanced competition. Well, let's start with, what's the problem that they're trying to address? So there are bugs like E. coli and Salmonella that are living in your gut harmlessly and not causing disease. But for whatever reason, perturbations in the gut, environmental cues, hormones, immunotherapy, immunosuppression, these bugs upregulate virulence factors that cause them to become pathogenic. And a lot of them are drug resistant. And there's an increase in drug resistant E. coli and Salmonella infections. And working in viral immunology, I always am appreciative when I jump into these papers of bacterial immunology that, man, are these bugs diverse. And it's very difficult to generate a vaccine against some of these intestinal pathogens because of their diversity. And the current vaccines we have, they're really, I guess generally, we think about this concept of preventing disease or preventing in colonization or infection. And so what this paper is trying to address is what are ways that we can better target not only, yes, the disease causing bugs, but could we prevent their colonization completely before they even become the bad guys? And that's a very different challenge. So their proposed approach of vaccine-enhanced competition is you're combining two different components. An oral whole cell inactivated vaccine that induces strong IgA antibodies against the pathogen. So this would be, and what we'll learn, T-cell dependent, stimulated B cells, memory cells that secrete IgA and can bind to the pathogen and eliminate it or prevent it from infecting. But that doesn't always stop colonization. It's not sterilizing. I don't even know if there's such thing as sterilizing immunity. So the combinatorial approach would be a live non-pathogenic niche competitor bacterium. So what's this idea of niche competitor is that you would have a bug that does not cause disease, but occupies the same metabolic niche. And it would be a bug that digests sugar similarly or needs the same type of sugar. Basically, that would outcompete the bad guys while IgA is also targeting it. And so their paper, they really in the introduction, this isn't always common in papers, but they provide you some supplementary materials in the intro before they even get to the main figures, where they present a mathematical model. And their model they grow different bacteria at different rates, and they have limited intestinal niches, and they incorporate those growth rates, gut flow, the death of the bacteria. They put all of this into a computer model, and they looked at what the model predicted. And so they found that it predicted two main things: how strongly do IgA antibodies increase clearance of the pathogen, and then how effectively does the competitor strain suppress the pathogen by occupying the same metabolic niche. So exactly what I had said before, that is what led us then to, okay, show us the proof of concept that this could work in mice. This is all done in mice. So the first thing they do is they take Salmonella typhimurium and they create an engineered non-virulent competitor. They delete key virulence genes to make it harmless, and they modify the O antigen on the outside so that the vaccine that you give against Salmonella doesn't removes the wild type Salmonella, but not the competitor, but the O antigen's gone. So what they did was they orally vaccinated and pre-colonized with this competitor, the niche competitor, before giving streptomycin treatment to keep out all the other bugs. And then they challenged with wild type Salmonella. And what they found is that vaccination alone or the competitor alone only reduced Salmonella wild type infection modestly. But when you combine them both, you had near complete clearance of wild type Salmonella. So prevention of systemic spread, elimination of intestinal inflammation. And what was interesting is they found clearance was equally effective if the competitor, again, this non-virulent Salmonella, was introduced days or weeks before the infection. So it didn't have to be exactly at the time of infection, which is really interesting. And so they asked that same question about the metabolic niche. So they took their modified competitor and changed its ability to metabolize a sugar. And they found that even that small loss in this ability for the competitor to metabolize the sugar, which also the wild type Salmonella needs, the effect then was lost. You really do need this overlap in the competitor's ability to take over the metabolism of the wild type bug. So that was with Salmonella, causes disease. They showed it works. They next wanted to ask if they could, because that was also a very artificial system, right? It's an engineered Salmonella to not have virulence factors in the O antigen, which in the wild would not. That wouldn't be possible.
And it's also was a prophylactic model. Right. So, the idea was that they were going to use the virulent Salmonella at the end. So, I know, for example, sometimes when I travel, I have to get a typhoid vaccine for Salmonella. And so this would be the same kind of thing of something you might do before you were going to be exposed to virulent Salmonella. But most of the time you don't know you're going to be exposed to a virulent GI microbe. And so I think the other thing that's really cool is they also think a lot about other models of what happens if it's therapeutic, what happens if you're already colonized.
Right. Yes. And I do like the idea of like, introducing, you do the vaccine, like you said, Brian, and they talk about this at the end. You take a probiotic that can take over the niche of the bad guy if the bad guy came infected your gut.
So I had one question, and I don't know if I missed it or I didn't get a chance to read it as carefully as I wanted to. But did they, they pre-gave this competitor, but do they actually quantify how much of that stays around over time?
These are short studies, and it was one of the limitations they mentioned that they don't. In any of the studies, they don't go past day 10 or really day 14 in their system. So they show, clearance or changes over time by day 10, but they don't show us longevity, which really would be key to understanding whether that competitor can stay colonized. Because I think there's a lot of literature that pre and probiotics, you can get temporary colonization, but they don't really stick around.
Yeah, because you could take massive amounts of those probiotics, and there, first of all, aren't very many live bacteria in it, and then second of all, they don't last
persist.
Yeah. So I just was wondering about that, because if their argument is that this competitor is truly out competing, how long does it stay around? How long before they challenge do they have to give it?
Right.
How quickly does it? Because are they also making space for it? So are they pre-treating with antibiotics before they give the competitor? Because another thing is if you just feed bacteria, they usually don't survive very long.
Yeah, they do make space for it by doing a streptomycin treatment before.
They do. Yeah. And those like first proof of concept studies, this is a very reductionist approach. They're really taking out all the potential confounders, which would be the other microbes. And so that's a really important point, that our guts are very diverse.
Yeah.
So they try to start working towards kind of this concept that, okay, let's kind of build in back some of these complexities. So they wanted to look at a commensal E. coli strain that has partial metabolic overlap with Salmonella. Now, what they found is when they combine vaccination with this E. coli 8178, it reduced the initial Salmonella expansion and prevented gut inflammation, even though it didn't induce an immune response itself, which is interesting. So again, it's about this partial metabolic overlap to outcompete Salmonella. So it didn't have to be this completely contrived or at least this engineered Salmonella. You can use a commensal that has partial overlap, and it seemed to work. They did a totally unrelated probiotic, Lactobacillus casei. It had no protection. So this niche similarity is really essential. They also did an interesting experiment where, we talked about how they had to make space by using an antibiotic. They wanted to do a model where the end result was a more intact microbiota, not, just stripping it with an antibiotic. They did this high fat diet perturbation. So feeding mice high fat diet does cause dysbiosis in the gut, without eliminating the bacteria in the way an antibiotic would. And what they found was the combined strategy of vaccination and the competitor did perform best, but it wasn't as distinct. You didn't have like these differences. It seemed like vaccination, the competitor did something, and the combined strategy was the best, but error bars are a little bigger. So it gets us into this, once you start expanding the complexity of the system, some of the effects may not be as strong, but I still think effective based on their data. They then wanted to know if you could do this if the bacterial strain was already established in the gut microbiota. So they used, a different commensal E. coli strain in the mice and allowed for persistent colonization without antibiotics over time. And then they gave an oral vaccine specific for that bug, along with a cocktail of competing non-targeted E. coli strains. So again, a commensal that already existed, you vaccinate against it and then provide competing E. coli. And this isn't a disease model, so there's no inflammation. They just looked at colonization, and they found that the combination of vaccination plus competitors led to complete elimination of the strain in 50% of the mice and major suppression in the rest. So again, not complete penetration, but overall reduction. Lastly, they wanted to know some of the mechanisms involved. And I talked earlier about CD4 T cells and T cell dependent IgA responses. You would assume these would be affinity matured, more able to strongly bind the pathogen. So when they deleted T cells, specifically CD4 T cells, and you saw that this effect went away. And so that helped them define this to be a T cell dependent response. And so it wasn't I thought the experiments were really interesting. I thought the paper was pretty digestible. And the overall kind of bottom line that you can come away from this work is that the three things have to happen. The pathogen has to be a bacteria that you want to eliminate, in this case, Salmonella or E. coli. The competitor has to be harmless, and it has to shame the same metabolic niche, at least partially, but escape vaccine-induced IgA. See, that's the other thing, is you don't want the competitor to also go away because IgA binds it. And this strategy is this like immune plus ecology approach. It would be an antibiotic-free way to change the microbiome in a way that prevents infection. I love the concept. I think it's super interesting. A lot more work to do, but it was an intriguing paper.
Yeah, I was super excited and wanted to be like, showing it off to all of my students and be like, look, this is why we make you take ecology.
So this could be useful for C. diff, right? Where not many people have C. diff, but then when they get antibiotics for surgery, then it goes wild. So you could treat them ahead of time. I'm not sure if C. diff is ever beneficial, so maybe it's not a problem if you take it away.
Well, and it's hard. So they do talk about C. diff. So they do talk about the problems with this approach. I mean, extensive strain level variation. You have to identify the most relevant vaccine combinations, and the vaccines might not always work. C. diff produces an S layer with a highly polymorphic exposed epitopes, which is just really challenging for vaccines. So it's kind of like, yes, vaccine plus competitor probiotic is great, but the vaccinologists are like, yes, but we still don't have a good vaccine. So you're still back to vaccinology and trying to.
Do you think it could lower the efficacy need of a vaccine?
Well, that would be, I think, the goal based on this work. Absolutely, is that
because if you have a crappy vaccine, but you can supplement it somehow, maybe that helps.
Yes, and I think that would be really interesting to take it into some of these pathogens like C. diff, Klebsiella, to see if you can see similar effects.
So besides Salmonella, E. coli would be another one, the enteropathogenic.
Yep.
Shigella, C. diff, Campylobacter.
Yeah.
These would all be candidates for that.
Yeah, and how interesting if you could find, if you had a multi-antigen approach where you could try to generate a response to all those things, and that'd be interesting.
Yeah.
And I love it also for agricultural species because of antibiotic resistance on farms and issues with not, if farmers use antibiotics, they can't sell the product until there's a withdrawal period. And there's a concern about just general antibiotic use in agriculture contributing to resistance in human health. So I think it has diverse potential.
Yeah, cows, pigs, sheep, goats, right?
Yeah, that's a great point.
Yeah, we always think of humans, but those farm animals. A lot of other things out there,
There are.
Okay, now Brianne will tell us how to live forever.
The billionaires are going to come and knock on Brianne.
They are. So this is a paper that came out end of January in Science Immunology. And when I looked at it, I was like, what? How? What? And so felt like it definitely was one that could get some discussion. It is also open access. So I know sometimes I struggle to get science immunology papers, but this one was pretty straightforward.
Too. Even at my university, I'm like, what is going on?
Science immunology can be a little tricky. So this one's open access. So don't let that scare you away. the title of the paper is B cells drive CD4 T cell immunosenescence and age-associated health decline. There are two co-first authors, Saad Khan and Mainak Chakraborty, and two co-senior authors, Sean Weiner and Daniel Weiner. And all of these folks are from University of Toronto or Toronto General Hospital. There are a few at some other research centers, mostly in Canada, though there are a couple outside, including someone in Bratislava, and someone who is in an aging institute in the US. And these,
you mean it's an institute that's getting old?
They call it the Institute for Research on Aging, but it probably is also getting old. Yeah.
Yes, aren't we all?
And so these authors are sort of big picture interested in thinking about this idea of why the immune system kind of works less well with age. And so we know that as individuals age, they have sort of decreases in immune function. That's why we often think about high risk age groups for different infectious diseases in the aged population, and think about different vaccines and all of those types of things. But we never totally understood what was going on with that. We've had some ideas of some contributors, but there's still always been a lot of open questions. We do know that there's sort of this chronic low-grade inflammation called inflammaging. And we know that there are some big changes in some types of immune cells. It seems like T cells have less sort of naive T cells, and we have an expansion of memory cells as you age, which makes sense if you have sort of this low-grade chronic inflammation. And if you have sort of, mostly your memory cells proliferating. It seems like there is also kind of a shift with B cells towards certain antibody secreting cells and age-associated B cells with increased inflammatory settings. But there's a lot that's really unknown about this process. One other thing that they mention in the introduction part that ends up coming back to be sort of important later is they do sort of mention that the differences in T cell aging are complicated because it looks like different things might be happening with CD4s and CD8s. And they just kind of talk about, well, there's heterogeneity there, so it makes it complicated. And they
That's always what immunologists say, right? When they can't explain something or they have different results, there's heterogeneity.
Yeah. So they're sort of like, there's heterogeneity between CD8s and CD4s. And they do kind of remind us, when they're talking about B cells, that B cells, of course, can present antigen on MHC class 2, and interact with CD4 T cells. And that's sort of a unique aspect of B cells compared to many other cell types.
Brianne, when we talk about aging, like when does that begin? Like at 35, 40, 40? What is? I know there's no cutoff, but
so they, I think people argue about that, and it sort of depends on what measure you're looking at. So I feel like here at some point they had mentioned something about, they mentioned the growth of the global aged population, and they specifically say that that's above 65 years. And they talk about changes in different non-communicable diseases.
It's good because at 65 you get a discount on the New York City subway.
Perfect.
Makes up for it.
Exactly.
You know?
Exactly. And so they sort of just talk about, the aged population here and don't really put a number on it. And I think I've seen different numbers in different places. In their experiments, they are going to use mice that are aged mice, and the aged mice are between 22 to 30 months old. And they often are 2 years or 24 months old. And they also, at a later point, talk about middle-aged mice who were 12 months old. But I don't know how to correlate those exactly to where we would be thinking with humans. But I definitely know from my experience working with mice that 2 years old mice are pretty old.
Yeah.
I think aged mice is funny. It sounds like aged cheese.
Yeah.
Oh, there's a chart. Yes. There's a chart for comparing.
I've heard of a chart and seen a chart, but I didn't remember it. What are you seeing?
Okay, it's too small. Hold on. Okay, so 28 days is about 6 months. 70 days is equivalent to 20 years old. 1 year, like around 1 year, is equivalent to 40 years old.
Okay, so that's their middle age in this paper.
Well, it's and 2 years is equivalent to 80 years old.
That's aged.
That would be their aged mice.
Yeah. So if they're taking them at, you said 18 months to 2 years.
So they say 22 to 30 months, but most of their aged mice are 24 months old.
Okay, so you're talking basically around 78 or older, according to.
Okay, and then when they do the middle age later, it's 12 months, so that's about 40.
Right. I think I would agree with that.
Yeah, I can live with that. So
do mice have also this inflammaging? And
yes,
they do. Yes,
chronic inflammation.
They do, and they're measuring that in a number of these experiments in this paper.
Right.
So in fact, the first thing that they do is they just take a group of female mice and they just measure their immune compartment when they're aged and actually document what the effects of aging are on B cells and T cells. So they sort of, go through and describe what they see in terms of B cells and T cells in their aged mice. And they note that basically what they see is in line with what previous observations had been. And it's a lot of the same kind of changes of more activated B and T cells and more of this sort of inflammaging.
Is there any sex-based difference in inflammaging?
I'm not sure. Here, they're only looking at females.
Yeah, I wonder. In mice or humans.
Yes, yeah, I'm not sure. And then they make this sort of turn in the paper, and I didn't know from my background why they would have gone with this experiment, but they did.
Some piece of prelim data, maybe somebody.
Yeah. I was sort of like, how did you get here? But they got here, and they decided to see what the role of B cells was in the aging of the mice, and so they made some mice that were lacking B cells, which people have made long before. These aren't newly generated mice. So these are mice that are B cell deficient, known as the mu-MT mice. So they have a deletion in the splice site that allows them to make a transmembrane version of the IgM B cell receptor. And so these mice can never make any heavy chain that can go on the cell surface. And so B cells die during B cell development. So these mice are completely without B cells. And these mice, like I said, have been made for have been around for a long time. But here they actually aged these mice to about 24 months. So, 80-ish or so. And then looked to see if there were differences in aging. And like I said, I have no idea why they decided to see what the role of B cells was in aging.
So I have two hypotheses. One is that somebody had some mice they forgot about.
Oh, that's
and they realized they were in the colony and they're like, okay, well, we have to sack them. But does anybody want to do anything with them? And then they realized what was going on. Or the other thing is you can actually get aged mice from the National Institute of Aging to do some studies, some preliminary studies and things. And maybe there were mice that were available and they were like, hey, maybe we saw some changes in B cells or whatever. Maybe we should check and see what happens. I don't know.
Yeah, they do somewhere mention like a little bit of data about CD20 that was out there before the paper. And so I was like, maybe that was also a hint for them. But it to me was all new information.
I love the idea that they just forgot and they got old. And then didn't we do an episode on I just remember someone describing a story to us, or maybe to me separately, where they observed a phenomenon when they accidentally let the mice get old. Yeah.
I don't
Mistakes can lead to good science.
So they looked at these mice that had no B cells, and they examined what happened in terms of the aging of the T cells in these mice. They used single cell RNA sequencing and looked at the different types of T cells in either the wild type mice or the mice that were B cell deficient. And they saw, and again, these are in the old mice, in aged mice. People have looked at B cell deficient mice for a long time. It's key that they're aged. And they saw that the aged B cell deficient mice had big differences in T cell populations compared to the aged wild type mice. In particular, the B cell deficient mice had far more naive T cells and seemed to have fewer sort of exhausted memory inflammatory type T cells. And these differences were more extensive in sort of CD4 groups, although a lot of what they were looking at here involved looking at sort of all aspects of T cells. And this is also all T cells in the spleen.
So were you surprised by that?
By which part?
Just that there's more naive T cells, fewer exhausted T cells. I don't know that I would have been, I don't know that I'm not surprised by that, because you don't have B cells to activate the T cells. So you just are going to have a less of a T cell response in general.
Yeah, I guess I don't know how much I had thought about B cells as the antigen presenting cells versus other cells as antigen presenting cells. I don't know if I would have said, oh, in the absence of B cells, there's going to be so much less antigen presentation to stimulate some of these T cells. I would have thought that perhaps the dendritic cells or macrophages could have picked some of that up. But
well, I think they could do the activation, but like there's not going to be germinal centers. Yeah. There's not going to be robust maintenance of the response.
I guess I hadn't, to be honest with you, I don't know. I thought about a lot of that until this paper. So
that's fair.
I don't know. Yeah,
that's fair.
But so they saw those differences. It seemed like the B cell deficient mice had T cells that looked sort of more of a young phenotype, while the wild type mice had more of the aged T cell phenotype.
And that's in the aged mice, but do we know that that T cell development and everything happens normally in the B cell deficient mice, like up to that point? Is this just like an aged phenotype, or is this just T cells in general are different?
So they looked specifically at the B cell deficient mice and wild types in young mice and saw no difference in the T cells. Mm-hmm. So they really only saw that difference in the aged mice.
Got it.
In terms of this T cell response. They also looked at VDJ sequences of the T cells to look at clonality, with the idea being that if there was reduced clonality, then perhaps that would mean that you had more naive T cells coming out of the thymus. If you had sort of the clonality was otherwise you would have more of the same clones proliferating. And you wouldn't have the same amount of diversity. And so they did see differences in clonality. They didn't seem huge, but I also don't look at these data very often to know exactly what kind of differences to expect. But the data that they saw sort of supported the idea that they were seeing less of this sort of T cell aging in the mice without B cells. And they didn't see that in the wild types.
And this was all, the differences were in the spleen when they looked in the thymus.
So they're in right now they're just looking at spleen.
Right. Okay.
And then exactly as you point out, one of their first questions that both you and Cindy asked is this a question of differences in the thymus and thymic output, or is this a difference in what's actually happening to the T cells in the periphery? Mm-hmm. So they measured the thymus in the aged B cell deficient mice and the wild type mice, and they didn't really see any statistically significant differences. And so it looked like the thymus and all of the aspects of thymic output were the same. And the difference was in the T cells in the periphery. Again, specifically, they're looking at the spleen here. And so they then did a lot more work in looking at transcriptional activity in those T cells to try to understand what kind of function those T cells had. And they saw that the wild type T cells that were aged had more of a sort of activation and inflammatory profile in terms of their transcript. And the B cell deficient aged T cells had more of sort of a stem and naive type of transcriptional profile in the T cells. And so this was all sort of, work coming together for them. And then they did another experiment, which I was pretty happy to see them do, which is that they also went away from looking at sort of transcript and actually took out some of these cells and measured their function. So they took the T cells out of the aged mice, and these are T cells that had either been in a B cell deficient environment or T cells that had been in a B cell sufficient environment. And they looked at the cell's ability to proliferate ex vivo, make cytokines ex vivo in response to PMA, make phosphorylated stats, proliferate with Ki67, do have sort of DNA damage markers with H2AX. And they saw that sort of in all of those cases, the cells from the old mice were sort of more inflammatory, more proliferative, making more cytokine, while the cells from the old B cell deficient mice were making less cytokine, less inflammatory, less functional in their functional assays. And in some of these assays, they also got B cells from young wild type mice. So they had B cells from young wild type mice, B cells from old wild type mice, or T cells from old B cell deficient mice. And they saw that the T cells in the old B cell deficient mice resembled the T cells from the young wild type mice. So it was as though the B cells had perhaps done something to the T cells in the older mice.
Oh man, it's bad B cells.
Yeah, I guess it's really hard to say whether the B cells were doing something bad or the T cells failed to do something in the absence of the B cells. So are they just not getting activated? And that's why they look, the younger ones look less activated and less mature and all of that, just because they haven't had the experience.
Right. Yeah. And from these experiments, they can't quite tell, is it that the old B cell is doing something bad, or is it that the T cell is lacking something? Exactly.
Yeah.
So Ben, have people done T cell depletion experiments and looked at this?
Not that I'm aware of. I think that I can think of so many questions coming out of this paper that I don't know the data on, but I can think lots of people can come up with lots of new cool experiments.
Sorry, Brandy. did you get to they put young cells in old mice?
No, I was
sorry. Okay.
Just going there. Yeah.
Yep.
Yeah. They also looked at things like an eye age index or a Sen-Mayo score to look at inflammatory age of the T cells. So apparently there are sort of scoring algorithms for this. Then before they do the experiment that Steph asked about, they have one other experiment that is also really interesting. And so they are showing in these previous figures that there are differences in T cell aging in the B cell deficient mice. And they wonder how that relates to kind of aging as a whole or aging in the organism and other things we usually think about in terms of aging. Because typically when one worries about changes associated with aging, their first thought isn't, oh, what's going to happen to my T cells?
Exactly. Maybe an immunologist might.
Yeah, maybe an immunologist might.
Most everyday people are more worried about wrinkles and gray hair.
Right.
Weight gain and heart disease.
So they actually just measure some of these other kind of health parameters in their B-cell deficient mice. They find that the B-cell deficient mice have slightly lower body weight, though it's not a huge difference, but they found a big difference in a 31 index frailty score. So the B-cell deficient mice are much less frail than the wild type mice. They also show differences in metabolic sensitivity with glucose tolerance tests and things like that. They live longer. So the wild type mice die quicker than the B-cell deficient mice. They have differences in secretory phenotype or in senescence related genes in the liver and in the lungs. They see the same kind of T-cell differences they'd shown before in places outside of the spleen. And they see differences in fibrosis as well as improved grip strength in the mice that don't have B cells.
Yeah, that's what I want.
So you can open those
the grip strength, it's all about opening the pasta sauce cans.
Yep. What's interesting to me is that we're like, the immune system is so important. Immune system fights disease. And now you're saying, oh, you don't even have any B cells and you're better off. And I think we have to be careful and make sure that we don't try and translate these data to humans and their everyday life, because these mice live in what we call SPF, which is very engineered system. They are drinking sterile water. They are eating sterile food. They have HEPA purified air. They are checked every day. We have what's called sentinel mice to make sure that there are not microbes in the colony. So it's a very controlled environment, and what kind of pathogens are allowed in those animals. So they're not experiencing colds. And I think, Steph, you were telling us that some virus went through your family recently, right? My son just texted me, he's sick, right? Everybody's sick, and we get these colds every year. And if you didn't have B cells, it's not going to be great. So this is not a solution in the long term.
Right.
Well, maybe there's a part of B cells you can remove and live longer, not the whole thing.
If we understood what part of it was important,
exactly,
and it wasn't critical for everything else, then maybe we could mess with that one part. Yeah.
And Brian, did they talk about the mechanism of frailty, like the CD4?
They don't really talk about what frailty. I mean, they talk a little bit about changes in the mechanism for changes in the T cell compartment, but how that leads to frailty or fibrosis or things like that.
We can assume it's like because this concept of inflammaging, it's like inflammatory cytokines interacting with muscle cells. When they're talking about mechanism at the end, there's a little bit of sort of discussion about some of that, but that part, because it was not my area of expertise, I was not totally certain about all the things that they were talking about there. Right.
So I don't know how certain the mechanism is, but I think that knowing this and trying to figure out what part of B cells is leading to this could be a really important avenue.
For sure.
And exactly as Cindy was saying, part of me wanted to know, and this will also come up with one of the later experiments, what happened if you challenged these mice with different pathogens? Would the T cells, these great healthy T cells they have, how would that help them? And what if you had pre-vaccinated these mice and then challenged them with the pathogen? If you vaccinated them when they had B cells, could they then be protected? And I guess that's talking about the fact that they're going to do an experiment later where they get rid of the B cells.
That's one way to live longer. Brianne, that's one way to live longer. Get vaccinated.
I know, right?
I was curious. I just looked up this 31 frailty score, index frailty score. Yeah. What might be interesting to some people is some of the markers on that are alopecia, so hair loss of fur color, so going gray, changes in skin, loss of whiskers, tumors, tail stiffening, gait disorders, balance, hearing loss, cataracts, vision loss, malocclusion, so tooth issues, diarrhea, breathing rate, temperature, body weight.
Yeah, you're describing aging, yeah.
Exactly.
There is actually a frailty index for humans too.
Yeah.
And we did a paper on Twin a while ago showing the microbiome correlates with frailty, So that's another thing here they could have looked at.
Yeah, absolutely. But since before I read this paper, the idea that B cells had anything to do with it would never cross my mind. It seems like
right.
But Brianne, that we know the immune system can be very dangerous, so it shouldn't be surprising.
If you had said macrophage, I would have been like, oh, absolutely. Macrophages do everything good and everything bad. No problem with that. But B cells, I didn't think they were that bad.
Yeah. So then they move into some experiments that kind of...
We'll never look at a B cell the same way.
They go into some experiments to kind of ask the question we were talking about before, where is it the old B cells that are the problem, or is it sort of, a lack of something in the T cells? And they address this in a few different ways. So one of the things that they do is they do a depletion of B cells in their what they call middle-aged mice. So I was incorrect. It says that they're 14 to 15 months old, not 12. So I guess they're older than 40 if we're putting them on a human scale. And they deplete B cells in these mice using an anti-CD20 monoclonal antibody once every 2 weeks for 12 weeks. And of course, that might pique some people's interest because there are some humans who are treated with anti-CD20 monoclonal antibody, though it may not be, as complete or as long-term as what you're seeing here. And so they basically look to see what would happen in the aged mice if they lost their B cells only when they were aged, but had B cells when they were young. And when they did those experiments, they saw that getting rid of B cells when you were aged made the T cells have the more young T cell phenotype. And so it seemed to be more the presence of the old B cells at age was causing the T cells to start to have this quote unquote old inflammatory activated phenotype. It wasn't, the T cells themselves intrinsically having an issue. And so this is, I think, the place where I was thinking about, well, what if you had vaccinated before depletion with challenge? I sort of wondered a little bit about that. And they saw that when they did this treatment, they saw the improvements in T cells. They saw the improvements in frailty. They saw sort of all of those improvements when they depleted B cells in middle age.
I find that kind of surprising because that is suggesting that there's something, actually a positive signal that the B cells are providing, that when you take it away, the T cells can rebound.
Yeah. Well,
and I'm kind of surprised by that.
Well, so it's funny because you're calling it a positive signal. I kind of think about it as sort of, there is a harmful signal the T cells are providing that are pushing that the B cells are providing that are pushing the T cells to this problem phenotype.
Right. That's what I meant. But when you take it away, they bounce back. That's the part that I'm surprised about, because I could envision the other way around. Like if you take aged B cells and you give them in, and that causes T cells to move to the aged phenotype. But it's kind of hard to get them back from that inflammatory phenotype to a quieter phenotype.
The thing they don't really show here is that when they do this anti-CD20 treatment, they're in mice that are 14 months old. Mm-hmm. And then they test the effective aging at 24 months.
Yeah,
so you don't actually know if sort of the B cells were already bad at 14 months or if you prevented them from going bad.
Yeah. And so that's probably more likely, right?
Yeah. And so if it's that case, then the T cells wouldn't have had to revert. they never would have gone bad in the first place.
Yeah,
and so we can't tell, was this depletion before the B cells went bad or after the B cells went bad?
That's fair.
They could do a time course at different times to see when, like, you keep backing it up.
Yeah, you could move to different time courses with those, taking out those B cells and see where the B cells go wrong.
Right.
Definitely.
And that's a tough experiment because if you have to continue to deplete for a year.
Yeah. There are so many experiments, especially in this sort of part of the paper, that are really fascinating questions, and the data are, not like super clean, but also if you spend some time thinking about it, you're like, whoa, that is a really hard experiment.
Right. And like eventually grad students have to graduate and postdocs have to move on. And,
Which is the cost.
Right.
And the cost. Yeah.
Well, because how frequently did they use the anti-CD20 depletion?
So it said they were doing it once every 2 weeks for 12 weeks.
And then not after that?
That is what it said.
And then they just look at them at a year because those B cells are going to come back.
That is
so there's something about the middle-aged B cells that once
time.
You change it, then it changes the trajectory.
Yeah, that's a lot of that they could one could look at in the future. You're absolutely right. So they then take out aged B cells and co-culture them with young T cells versus old T cells, and show that it is not the old T cells that are the problem. It is the old B cells, because if you put old B cells with young T cells, the old B cells actually change the phenotype of the young T cells and make them act more like the inflammaging T cells.
I wonder if this just can be that older B cells are making more cytokine, and then you're just antigen independently causing T cell changes.
Yeah, no, I think that very much could be a part of this.
And it's very rational. It would be something like, B cells also get old.
Yeah, well, I think it's interesting that B cells get old, but maybe the T cells aren't having the same issues.
It is interesting that like the T cell immunosenescence that we talk about, it's almost like they're like, it's not our fault. It's all these other actors acting upon us. So if you just left us alone, we'd be fine.
I wouldn't get so old if all the people around me didn't cause me to get old.
And then they did the experiment Steph mentioned where they took the aged B cells and they transferred them into young mice to see whether they would get the aged phenotype if they put aged B cells into young mice. And again, they saw the same kind of aging phenotype if they put aged B cells into young mice. So again, it really looked like that's something about old B cells that are causing the problems.
Now, they didn't do the young B cells into old age mice because they kind of already did that with the middle age experiment, right? Like they didn't really need to do the reverse.
I guess they didn't need to do it. I don't really know what their decision process was, but
I mean, that's the experiment that'd be great, right? Because that's like the fountain of youth experiment, right? Like, so if you can take young B cells and put them in an old mouse and they get better, then that'd be great. But that's not the way this works. So you got to go and deplete something.
Yeah, the problem would be you'd have to get rid of the old B cells.
Yes.
Right. So the question would be, in a B cell depleted old mouse that you have like they didn't grow up without B cells. You deplete them when they're old, and then you give young B cells. That's the experiment that I would have liked to see.
Yeah. So they didn't do that, but that could certainly be a cool thing.
Yeah. Yeah. And it just reminds me of like these, once in a while, these articles I see of like some older billionaire, like harvesting the serum from his son and like transfusing himself with his son's serum. And
Yep.
Yeah.
Maybe you could like save some cord blood when you're born and use that to give yourself some new B cells.
Like biobanking your stem cells.
Right.
That's interesting.
Yep. So now they want to know what it is about the old B cells. And they kind of want to look into the mechanism. And they spent some time talking about insulin and data on insulin's impact on B cells. And some of it I got a little confused by because it felt a little circular. But overall, what they ended up doing to experimentally look at this is they made mice that they first analyzed some data on young and old B cells that was just publicly available data. And they saw that there was a big difference in terms of insulin genes in the insulin signaling pathway. So it seemed as though old B cells had more going on with insulin signaling than young B cells. So they looked at that, and then they made mice where they did an insulin receptor knockout in just B cells using Crelox technology. So they were able to make the mouse was insulin sufficient, had all of its sort of normal metabolism, but B cells themselves did not have insulin receptor and could not respond to insulin. And if the B cells can't respond to insulin, then the B cells actually do not get this aged B cell phenotype and this sort of problem phenotype. Again, they talk a little bit about, whether the B cells are hogging insulin from other cell types. And they talk about sort of this B cells get insulin signaling, and that changes the B cell and that increases their insulin receptor. And like, I got a little circular, and I need to talk to an endocrinologist to help me fully understand what's going on. But it seemed like B cells receiving insulin signaling was sort of a key piece to this. Something about the old B cells receiving insulin through insulin receptor was leading to this impairment. They also related to some of the things, and again, they showed that this involved the T cell changes and the frailty changes and all of those different types of experiments. And then Cindy kind of talked about how much this is related to B cells activating T cells and things like that. So they also looked at B cells that did not have MHC class 2, so that those B cells were not able to stimulate T cells. And they saw that, again, if they didn't have MHC class 2, again, they lost some of this phenotype. So they didn't see the sort of T cell aging phenotype as dramatically without MHC class 2. And in particular, they didn't see the aged phenotype as dramatically in the CD4 compartment. And so this would seem like it's something about the class 2 signaling to the T cells.
Yeah, but then that's circular, right? Because if the B cells don't have MHC, then they're not engaging the T cells and getting the signals from the T cells that might be necessary to produce whatever factors there are to age the T cells. So
to then affect the T cells.
Yeah. So you still, I mean, the experiment is good, but I don't think it really, I don't know. How would you ask that? Maybe... I don't know how one would address that. I'm trying to think, could you use like antigen specific T cells and specific, I don't know.
Yeah, I don't know. I think
I'd have to think a lot about that.
I think that all of this sort of, there's something with insulin, there's something with MHC class 2 in terms of mechanism, but it really doesn't tell us exactly what these B cells are doing.
Yeah. But
it does tell us the B cells are doing something that seems to have a decent effect on the aging phenotype in these mice. And if we could figure out that mechanism, which I, can imagine lots more challenging experiments that involve a lot of like aging mice, which is expensive, and depletions and transfers, and all of this are really hard experiments. I think it opens up a lot of really new and interesting questions about aging that I'd never even thought about before.
Yeah, there has to be some low-hanging fruit, though, because they've done some single cell, and I'm sure people have looked at B cells in the older mice. You should be able to see whether there's a secretome that is different.
Right.
And could you recapitulate that just by taking mice and injecting them with that combination of things and seeing whether that, causes the same phenotype? I don't know. Because then you could go back and then start knocking things out in the B cell and see whether that has an effect over time.
Right.
Yeah. There are people who are treated with anti-CD20. But are
they outliving all,
I don't know of any data on that. But I also know that generally people are getting treated with anti-CD20 when they already have other health conditions. So it would be a bit of a confounding factor given the other health conditions those individuals have before they get that anti-CD20 treatment.
Which are mostly lymphoma.
Yes.
and then also more recently, potentially autoimmune disease.
Right, some autoimmune disease.
Crohn's.
Yeah.
Right. But it's interesting when I was looking at T cell related phenotypic changes on people with rituximab, and they do have less inflammatory T cells. I'll try to find that.
But how is their grip strength?
Not great, because they're probably on rituximab because they have lymphoma or
rheumatoid arthritis.
A great question. So there seems to be some human evidence. It's surprising they didn't mention human.
They sort of really briefly mention something about, there was some data about anti-CD20 treatment. Okay. And a citation, and then they just moved right on and didn't really get into it. And I didn't know whether that might have been the reason why they looked at B-cell deficient mice in the first place, because that came out of nowhere for me. But I think it's really cool, and I'm excited to know it. I want to keep my B cells. And I do think that looking at what would happen with these mice if they had, a normal microbial compartment, or if they were challenged with certain things, or if they were vaccinated and then challenged with a depletion of cells and things like that, could be really cool things to see. Are there ways we can keep you healthy and modify your B cells? But I think the most promising part would be to figure out what is actually going on in those aged B cells, and be able to modify or modulate that. I also think it's sort of interesting, and Cindy kind of mentioned this, to think about evolutionarily what's going on here. I mean, maybe this is happening so late in the individual's life that it's not super relevant for selection. It's just that the B cells start needing insulin and having the insulin receptor. But it is sort of interesting to think about, do you need to stop using the resources of immune defense at some point, or just what's going on evolutionarily? It's interesting to see this as like the B cell is actually doing something, and that makes me have a lot of evolutionary questions.
It's interesting. I don't know how humans are didn't evolve for most of their existence to live so much, mostly 30, 40, right?
Right.
So this is probably not something that was meant to be, because you would never reach that age, right? And I don't know about mice. I'm not sure. Mice probably live the same forever, right? Except when you have them in a lab, they can live longer because they don't get eaten. But that's different, right? So I don't know. The evolutionary implications are different.
Yeah, well, I sort of wonder whether B cells, using insulin in middle age evolved for one reason, and then it went awry as you got older.
Well, you would usually die at middle age, and that didn't matter. So the late age implications were irrelevant if you died at 30 or 40. So and now we're living longer, and the other parts haven't had time to catch up, modify, and so forth. By the way, mice. So you said mice, 2-year-old mice are like 80 years in humans. Yeah. So these guys live 3 years. How much would that be? Like 120 years?
It's old.
Yeah.
Wow. Jeff Bezos is going to be all over this.
I mean, he just has to get rid of his B cells and then also never see any microbes.
We should just stay inside all day too and not see anybody.
Yeah. Well, so that is another piece of this, right? Because we house mice in conditions which they aren't normally exposed to.
Yeah, so a wild mouse probably lives a lot shorter.
But also a very different life, right? So maybe shorter because they get eaten, but as long as they have access to food, they're more active. They live in larger colonies. So their temperatures are different. There's a whole bunch of things that are different.
How long a wild mouse lives? There has to be. Let's look it up. How long do wild mice live? 3 to 12 months. 3 to 12 months.
Wow.
It's a lot less.
A lot less.
High predation, harsh environmental conditions, and lack of veterinary care. Hey, that's why humans live longer, because we have medical care, right?
Yes.
It's totally it. I'm reading a book by Neil deGrasse Tyson, and he's talking about this. He said, in the old days, humans ate free-range meat. They ate, organic everything. They had fresh water, and they still died at 30.
That is right.
So medicine makes a difference.
Yes.
Yes.
I've seen him say that before. Yep.
Yeah, it's really good.
It's right. I mean, people are trying to say, let's go back to the way it was. It's so much healthier. Like, oh, really? Was it though?
But you could drink your unpasteurized milk. Go ahead. I'm not going to drink it, but you could do it. Go ahead.
Okay, but then the people are feeding it to their kids, and the kid doesn't have a choice in this matter.
No, the kid is the problem, right? I feel very badly for the kids.
When you're making those medical decisions that negatively impact a child and potentially kill them, that is a different, I think.
Yeah, I agree.
That is why we have laws for parents not being able to neglect or abuse their children. Yeah.
It should include not vaccinating them.
That is in a lot of
great
cases, right? That happens in the court
system.
system.
The guy in Texas whose kid died of measles, he said he would do it again with another kid. Mm-hmm. I mean, you should go to jail for that. It's not being a parent.
It is one of those things, though. Like, it's so hard. I don't know who has gotten. I mean, they must have just been surrounded by, misinformation and still trying to deflect. I'd imagine, because it would be incredibly painful to admit that, like, your decision is the reason why your kid died.
Yeah. Oh, yeah.
I don't know. I would say I screwed up. I wouldn't do it again.
I think that you're not the norm in that, though. I think people would have too much pain around that they would need a psychological way to deflect that pain.
But all three of you would do the same.
Yeah.
Wow.
Right? Because you're informed. You're informed. Yeah. Well, you would never have done it in the first place. That's
the problem. Right. So it's hard to imagine.
Yes, it is hard to imagine anyone who would do that. Thank you, Brianne. Thank you, Steph.
Yeah.
That's Immune 102. You can send us questions and comments to immune@microbe.tv. If you enjoy these science discussions, this is just like a little journal club that we have here. I love it. You can get 4 people and you talk about a paper and get all kinds of ideas coming out. It's just great. If you like these, please support us. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Blue Sky. Thank you, Cindy.
Thank you.
Steph Langle's at Case Western Reserve University. Stephanie Langle on Blue Sky. Thank you, Steph.
Thank you. It's great.
And Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.